but let's start. So hello, everybody. Welcome to the Paper Trail. My name is Bria Macklin, and I'm a postdoc at the Gladstone Institutes. And the Paper Trail is an online interactive journal club that was developed by 4 former colleagues turned lifelong friends. So myself, Franklin Hall, who's a graduate student at Johns Hopkins University, Dr. Quentin Smith, who's an assistant professor at UC Irvine, and Dr. Michael Blatchley, who's a postdoctoral fellow at CU Boulder, and Michael's going to be your host today. So born from our love of discussing science, the Paper Trail, born from our love of discussing science and our yearly paper a day challenge, we came up with the idea for the Paper Trail to be a platform that allows for primary authors to come and share the story behind their research. And so we really wanted the Paper Trail to stand out from other online presentations. And to do this, we came up with a set of guiding questions that prompt the author to share more than just the results. And so we're super happy that you've decided to join us for this live stream. And in true Twitch form, we ask that you use the chat feature to ask questions throughout the presentation. And we have moderators in the chat, or the other hosts will serve as moderators, and they're going to relay your messages. And so please do not hold them to the end. Please ask your questions as they pop into your head. And right now, if you're looking at the video and you don't have access to the chat feature, what you can do is you can just really quickly create a Twitch login. It just takes a few seconds, and that will give you access to the chat feature. And with that, I would like to introduce our speaker for today, Dr. Kacey Ronaldson-Bouchard. And so Dr. Kacey Ronaldson-Bouchard is an associate research scientist at Columbia University's Department of Biomedical Engineering in New York City. Her research focuses on engineering multicellular systems from pluripotent stem cells, including the advanced maturation of iPSC-derived cardiomyocytes into mature functional cardiac microtissues. Dr. Ronaldson-Bouchard has interest in integrating multiple engineered tissue models for understanding inter-organ interactions towards the development of patient avatar models for the realization of personalized medicine. She has published papers in multiple journals, including Nature, Cell Stem Cell, and Cell, and is a co-founder of Tara Biosystems, a New York City-based startup company focused on engineering advanced cardiac models for drug discovery, testing, and precision cardiology. So we're super happy to have her here presenting with us. And so with that, I'll let you take it away.
Thanks so much. I am so excited to be here, and I really love this initiative. So I'll start. I need you to allow me to screen share. There we go. I'll do the typical Zoom. Can everybody see my slides? Yep. Okay, so here I'll talk about this paper that we recently published, this multi-organ platform with tissue-specific niches that are all linked by vasculature perfusion. So the acknowledgements, there's a ton of authors on the paper, but I really wanted to highlight some of the main people from the beginning to the end that made this work happen. So my PI, Gordana Vanjuk-Novakovic, saw the vision of tissue chips. She's been developing bioreactors for years, and so tissue chips kind of fit in really nicely to that paradigm. All of the technology has been developed by our in-house engineer, Keith Yeager. He's just a brilliant, brilliant engineer and worked so hard to bring, everything we have to life in all of these bioreactors. Diogo doing the PK/PD model, and then the rest of our team in different roles. We also outsourced a lot to cores. And I really can't, not thank our funding, because you'll see later that the NIH really drove this initiative. So some background, something you all, I'm sure know, drug testing is not predictive at all. So preclinical, you'll do some in silico 2D models. Recently, you've shifted into 3D organ on a chip models, and then everything goes through animal models. And what ends up happening is you have 10 years to get a drug to market, you're spending billions of dollars, and a very, very low success rate. And of those drugs you do get to market, so these are the top 10 best-selling drugs, at best, they work for a fourth of patients. So you're spending all this time and money getting a drug to market, and then it's barely working to, improve the lives of people. So it's pretty clear that preclinical research needs innovation. We need to speed up this timeline, and there's this unmet need to really parse out, how do we know what is working preclinically, if it'll work in humans in that clinical trial? And we saw that as a nice fit for organs on a chip. So having a human model as this missing puzzle piece, not always a replacement, but just, a tool to give you more insight into the mechanism or if there's a human interaction issue with your drug. And just a little more intro about organs on a chip. So it's not the whole organ in a Petri dish. You're really defining pretty much one functional unit of that organ. So for the heart, something that is conductive and contracts, so a ventricle strip of tissue, to do a very mechanistic study. So having a very controlled environment where you can start to parse out why things are happening and different mechanisms to get an answer. So you're not really trying to create everything, just a simple model to answer a very specific question. A little timeline about the work. So we really started back in 2010, and I'll show you why later. But as far as the multi-organ platform started, this version was in about 2016. And we did all these experiments. We picked other drugs, but 2017 and 2018, we were really doing some good runs and getting great results. Thought we were ready to publish, and then decided to have a higher N. And so trying to do that with maternity leave kind of delayed things, and then COVID happened. And so we did revisions during COVID, which isn't all that fun, but we made it happen. And we're in New York, so COVID hit us pretty hard. So it did take a bit to get out there, but I'm glad it is finally out. Did I freeze? Am I back? Okay.
All good, yep.
So what motivated this work? So I can't really tell this story without telling the story of tissue chips in general. And so in 2010, the NIH and FDA funded this tissue chip award to advance regulatory science. And then the next year, they had this workshop led by NIH, FDA, and DARPA, and really Dan Tagle at the NIH to make this coordinated effort, because they could really see the vision that tissue chips, or organs on a chip, MPS systems, there's been a lot of terms for them, but organs on a chip kind of stick out the most. They really saw that this could be a good tool to get drugs to market and take that burden off of the healthcare industry spending all that money to get a drug to market. So in 2012, they launched.
Question about that, if you don't mind. So that seems like a pretty fast turnaround from kind of the initial papers about tissue chips. Was there like kind of like a meeting about this to get this funding initiative, or were they just like really on top of it in terms
really
of,
on top of it? So tissue chips, I may be wrong. So the Science Lung Organ on a Chip Dan Hug Ingber paper is 2011, I think. But before that, you had Mike Schuler's work, Roger Cam's work. So not necessarily calling it organs on a chip like that pivotal paper, but just using microfluidic tissue chip systems. As it was more like microfluidic systems for drug science. So there's a big debate on who was necessarily first, but for sure that lung on a chip science paper, which may be 2010 or 2011, I'm sure everybody knows this paper, really sparked the field. But even before then, people at the government, mostly Dan Tagle, saw that this could really be a thing and started getting the funding together, having these workshops. And so you're getting all these tissue engineers who are, and microfluidic chip people who are very good at making tissues or building bioreactors in a large scale. And the government put a lot of money into tissue engineering, but we weren't really putting tissues into people, right? We were making very small tissues. And so the pivot on that is tissue chips. So we were good at engineering tissues, just not for regenerative medicine. So this was kind of a pivot that helped everyone. And so then in 2012, they actually launched a whole new agency with this funding. So NCATS was launched, and then the Tissue Chip 1.0 was born from that. And so at first, so these are 5-year grants. So the first 5 years were using tissue chips for drug safety. So is the drug safe in a human context? And then you have other funding initiatives, chips in space. And then the 2.0 version were disease modeling. So moving away from preclinical talks to, will this drug work in humans? Will it work in this disease context? And then just a couple of months ago in 2022, we had the first MPS World Summit Conference. And so it's kind of a true establishment of the field. And now you can, get grants all the time for this kind of work, but it's been like a decade in the making to establish the field, and more for, the pioneers of the field. But it's really been driven a lot. So you had the government really funding it. a bunch of tissue engineers really looking for a way to put their tools into use. And then you had stakeholders, so the FDA right from the beginning, and the IQ consortium, so a bunch of pharma representatives telling you exactly what they would want from these systems to use it. And so that was really nice to kind of work so hands-on, like every month giving updates to these people on these projects, because people actually wanted them. So building tools that people would actually use. And so our Tissue Chip 2.0 goal was so this is all disease modeling efficacy, develop a patient-on-a-chip model of a systemic disease. So we were looking at doxorubicin-induced cardiomyopathy. So our design goals were that it be patient-specific, so everything from one source of iPS cells, especially so you can model specific genetic mutations, having a high biological fidelity. Because we're using iPS cells, we need them to be mature. They have to talk to each other and connect, but still be stable. And then we need to get information from the system. So having online readouts, having it be modular and reconfigurable, so you can kind of use it for other use cases, and then adding on complexity as needed. And so we always use stromal cell cultures, but as these tools advance, you want to add in innervation, the immune system, other different modalities. So making it a system that is easy to evolve as the science and tools evolve.
So were these goals basically built upon like the Tissue Chip 1.0, like interactions between the funders, stakeholders, and the scientists, or is this like all your guys' idea and you kind of implemented it?
So the Tissue Chip 1.0, which was more toxicity screening, that had, we kind of had mini consortiums based on our organs. And so like the heart, we all had reference drugs and reference endpoints. So those design requirements were very standardized based on feedback and agreed upon. But then once we moved into the disease modeling efficacy, we were able to define our own because our disease models were so different. And so the main things were, not to use PDMS, not to use Matrigel, be all human. But other than that, we were allowed to define our own. So these are the ones that we defined for the systemic model need. And so other, people in this grant don't necessarily need to use iPS cells because they can get human cells that are primary from all the organs, but we're using like the heart muscle where you can't get primary cells at all. And so it's kind of a give and take based on what your specific question is, which is, nice that there's that flexibility. So we are using all the cells from one patient, so that gives you, tons of differentiations, getting these main cells, getting the stromal cells. And the main thing that you need to watch out when you do all these differentiations is that you get a very immature progenitor cell. And so you're patient specific, but you need to have a high biological fidelity. And so you have to differentiate the cells and then mature them even further because we want them to be physiological. And so a kind of a case study is looking at cardiomyocytes. So if you expose adult cardiomyocytes to isoproterenol, they'll increase in force and they'll increase in calcium. But if you expose iPS-derived cardiomyocytes, you don't really see that increase in force and calcium. And so we, published some result. This came out of the Tissue Chip 1.0, was really making a mature heart model. And so we found that taking these early stage iPS cells, making them into a 3D tissue, and then training them with electromechanical stimulation at an increasing regimen. So 2 hertz, 2.33, all the way up to 6, made them rapidly adapt. And so now if we re-expose them to isoproterenol, those mature cardiomyocytes will give you that increase in force and that increase in calcium. So now we've got that mature individual tissue. I've only matured one tissue. If I want to connect them to the others, I don't want to sacrifice, that's 2 weeks of differentiation plus 4 weeks of maturation that I just spent to get a mature cardiac tissue. I don't want to sacrifice that just to connect the tissues in, let's say, a common media. So I want to connect them in a way where they can talk to each other, but they still are mature and maintain that phenotype. And with cells, that phenotype is mostly controlled by the things in the media. And all the different organs that you're going to connect will have different media. And so our platform was designed to enable this connection of multiple tissues. And then also we focused on engineering all these mature tissues separately and then putting them in that platform. So the strategy was to kind of take a note from your body. So in the body, your organs are all in their own niches, but they talk to each other through your vasculature. And so we just unrolled the vasculature and put it on the bottom. So kind of simple approach where you have each, because you have a vasculature barrier, it gives you some media separation. And so you can have the tissue niche up top, but they can talk to each other through this recirculatory loop. So this is what the platform looks like. It's just size of a glass slide, clamps together. We put sterile gloves on to do this. And then you've got these modular transwell inserts. You'll plug and play the tissues that you want in the top, and then the system runs. And you can do different configurations. 1 tissue, 2 tissues, 4, whatever your heart desires. This is from the bottom view. So we're linking it through the vasculature where you've got this endothelial line channel perfused with endothelial cells, and you're circulating immune cells within that. And then tissue specific media is added to the top once you mature the vasculature. And then our reservoir is open to act as a bubble trap, and pretty much everything is open so you can sample it. And you don't really have this closed system where you have bubbles. So step 2, engineer the tissues separately and transfer them. So I'll go through how we mature each tissue briefly, and then we'll go through the validation of the platform. So these are what the tissues look like. They're pretty large. So a lot of organs on a chip are more 2D barrier function tissues that aren't very 3D, but we are tissue engineers in our lab. And so we did what we knew, and we wanted to make, kind of keep those 3D tissues, because especially as we were going down in size for some of these tissues, we were losing functionality. So the heart, we take cardiomyocytes and fibroblasts, put them in this pillar, this hydrogel around these flexible pillars, and then ramp them up in electrical stimulation to get nice cardiac striations. They can electrically capture at a higher rate, and it takes less voltage to do so. And then as you add more calcium, you see expected increases in force. For the liver, we also use stromal cells, so hepatocytes and fibroblasts. We'll spin them down in an aggregate plate to make a bunch of 3D aggregates. And then we take all these aggregates and we put them in a bigger fibrin gel. So having the aggregates keeps the hepatocytes functional, and they'll continue to secrete albumin, and then putting them in the fibrin just makes them into this tissue entity. The bone is an actual piece of bone from a bovine scaffold that we mill into a certain shape, and then we decellularize it to take all the bovine cells out, and we put in human cells. So MSCs that eventually become osteoblasts. So those are your cells that form the bone, and then monocytes become osteoclasts, your cells that resorb the bone, eat away at the bone. And so you've got, your bone volume can actually change with your osteolytic and osteoblastic cycle, and it'll reform over time. And then skin, you take fibroblasts and a collagen hydrogel just on your normal transwell approach. It will condense and form your dermis, and then you'll put keratinocytes on top. And as soon as you expose them to that air liquid interface, they just, cells always know what to do. That's kind of the approach between all these tissues as well. Put the cells in the right context, and they're the true engineers. We're just providing, something for them to sit in, but they ultimately know what to do when given the right cues. So if you expose them to this air liquid interface, it'll stratify all on its own, and then you get epidermis and a dermis with really nice barrier function. Our vasculature, we use typical transwells in our custom insert, and we basically put endothelial cells and then MSCs to function as parasites on, and we ramp them up slowly to a shear stress, so 0.5 dynes up to 1.88, and they'll, flatten out and really form a nice barrier. We've also looked at other pore sizes, so our goals were to, maximize the amount of cells on the surface and not have it, and minimize plastic being the transwell. So while 5, 10, and 20 micron pores are all great, 20 microns was that sweet spot where we had, no holes and really good barrier function. As you get larger into 40 microns, you start to get gaps and your functionality goes down. And so 20 microns was a nice sweet spot where we thought we'd have the most just cell-based barrier. Then looking, does the platform, okay, we built this platform, but is it worthwhile? Or do you want to ask any questions?
Yeah, I was going to say, I have a few questions. This is super interesting, just seeing all of it kind of put together. So I guess maybe first, just like a high level question about how did you determine to use those for tissues? Of course, there are other tissues and organs. Is that just, I guess maybe just what was the rationale for those?
It's a bit of a mixture of what we're good at and know how to make, and what our goals were. And so screening for doxorubicin and cardiotoxicity, we wanted the heart, You always want the liver because it's metabolizing the drug. The skin lets you both look at things like peripheral neuropathy or drug delivery through the skin versus the IV. And right now we just have a bone, but in some of our other models, we look at bone tumor or metastasis to the bone. So the bones are really nice, bulky, tool that's useful for cancer. But right here, it's just bone, no tumor. And our lab is very good at engineering bone. So,
Sure. Yeah.
And immune cells are kind of always thrown into the mix as best as you can.
Yeah. Have you added in other components, or it sounds like you have some other projects going on, like kidney tissue, for example, or anything else like that?
We've done other people in our lab look at neuromuscular junction. Then we'll give the platform to other people to use their system in ours. But we kind of try to not make model systems that we have, especially because you have to do all the differentiation since we're doing all IPS. So if it's not our area of expertise, we try to stay away from it or invite people who know what they're doing just to put their model into our system. So you can plug organoids in, you can drop really anything you want it into. It's basically treat it like a transwell.
Yeah, that makes sense. That actually kind of leads into my next question. So it sounds like people have come and you've also sent this to people. At what kind of, do you just kind of design a specific, it looks like each of the tissue kind of chips have a different design, of course. So do you just send them something that they think will work well for their specific tissue, or do you kind of have like a back and forth? Because it seems like a lot of the kind of tissue engineering, like materials approaches, are kind of, especially for like the heart, for example, you used what you did specifically for the heart, adapted it to the chip. Is that kind of the approach for the other tissues as well?
Yeah, and so we'll do a lot of projects in the lab where people will come to us and like, this is what I want to model, and you build them the bioreactor just on that specific organ and those constraints, and then you would mature the tissue and just transfer it over. So that base platform of just the tissue niches and vasculature stays the same, but then your inserts all change. And that'll definitely change based on the models. We want to never like simplify the model system just to fit our workflow. We would rather, keep the science as, best that it can be at that moment, and just figure out how to connect them.
Got it. That makes sense. And I'll maybe just ask one more so we can keep moving. So all of us have some background in sort of vascular tissue engineering, vascular biology. So we have a lot of questions about the kind of blood vessel component, but have you looked into like tissue specific vasculature at all? Or do you think that would play a role, or is that kind of like a future direction for this?
Yeah, so we'll get into it a little bit later. We see some hints that just if you put in regular endothelial cells, they'll take on their own specificity. And then we have been looking at tissue specific vasculature. And we've also tried other I'll go through the failures of why we're not, vascularizing every tissue and why that got too complex. But yeah, especially, the cells in the right context, they kind of take on the phenotype on their own. And so we've been, focusing on what's a good way to characterize that they are tissue specific. So it's not really defined for, like liver is a bit easier than the others.
Makes sense. Great.
So validating the platform, we needed to make sure, do they talk to each other? Can we maintain them for the long term? And then do they show tissue specific responses to injury? So if I do something to one tissue, am I damaging everything? And then their use case for drug modeling, so PK/PD and clinical validation. So looking at interorgan communication, so we wanted a ubiquitous marker, and that came from one of the reviewer comments. How do you know that something secreted in one tissue is actually getting to the other chambers? And so we were tracking things the cells secrete, but especially with stromal cells, it's kind of hard to pin down, I know the cardiac tissue secreted this, et cetera. And so we made cardiomyocytes with a GFP label on CD63. So all the exosomes secreted by the heart were labeled green, and we can, track where they went. And we see that they're, majority in the heart, but they are going to those other tissues. So they are communicating with one another. We looked at it for the long-term culture. So we looked at 3 conditions. So the inner organ, so with that vasculature barrier, so you have the niches, a mixed condition where everything's the same, but we just don't have an endothelium on that barrier. So this will mix all the medias together and be more of a common media approach. and then isolated, so no connections at all, just them in their own, each tissue in its own environment. So looking at them for 4 weeks, we see that the interorgan, really maintains the individual tissue striations in the heart, albumin in the liver. You've got your epidermis intact in the skin, whereas in that mixed media condition, you're losing your heart striations. You've got calcification in the liver, ossification in the bone, and you just broke down your skin barrier. So it's really not ideal for the long term for these tissues. Vasculature niche is maintained. You'll see our liver ECs are a bit leakier, which is common for liver endothelium to be leakier. Sinusoidal cells are. Our immune cytokines, when we culture immune cells in the presence of the vasculature barrier, their baseline supernatant and secreted factor stays pretty stable, whereas in the mixed and isolated, they just drop off. We also were able to maintain tissue-specific function, so the heart shows similar contractile from the beginning and to the end, except in the mix. The albumin production is the same in the liver, except for the mix. In the bone, trap activity, so your osteoclasts that are active only in the interorgan condition, not even in the isolated, which was really cool to see. And then in the skin, the same thing, you're maintaining that epidermis when you have the interorgan condition and losing it in the others. We did global proteomics and looked at shared pathways. And so these are proteins that are expressed in all the tissues. And there shouldn't be that many, expressed in all the tissues besides, basic proteins. And we see that in the mixed condition, there's actually quite a lot. So these are expressed in the heart, liver, skin, and bone. In the interorgan, they're mostly related to, cell migration, different cell cycles, the same in the isolated, mostly related to metabolic processes. So those should be shared. But in the mixed condition, we see things related to vasculature, cardiovascular system development, skin, ossification, TGF-beta. And so the media is, probably the cause and other secreted factors of the heart showing ossification or showing skin morphogenesis. And the same as like these organs should not necessarily show that, right? We also took this proteomic and compared it to adult tissue. So using the human proteome atlas, we tertiled, so first we looked at how the genes were expressed and if they shared expression with the adult tissues. And we found that for the most part, they really did. One caveat is the bone, is we're comparing to bone marrow here because bone's a really rare sample, actually. And then looking at how they're expressed. So we tertiled them into low, medium, or high. And we found that they correlated the best in this interorgan group. And then if we zoom in even further and we look at the top tissue-specific proteins for each tissue, we see that the interorgan group is really much more like that adult counterpart, even more than the isolated in some conditions. And so having that tissue niche separation was really critical, and you're losing all that tissue specificity with this common media or mixed media approach. If we look into what's...
Sorry, I've got a quick question about the expression correlation. So I want to make sure I'm understanding this right. So so they're still not exactly matching the adult state. Is that correct?
Yeah.
Okay. So is there kind of like, I guess this is maybe not that well defined, but maybe in these kind of funding meetings, is there something that if it's close enough to adult, you can consider it adult for this type of disease modeling? Or is that something that as kind of the non-chip technologies improve, you'll kind of incorporate those there and just keep improving them kind of in parallel? Does that make sense?
Yeah, definitely improving them in parallel. So we're always working on increasing the physiological relevance and the maturation. Always, it's especially working from pluripotent stem cells, you're missing, years of development. And so there's never really a true overlap. But typically in the organ on a chip responses, we're looking at a functional measurement. And so as long as the functional measurement is there, you can kind of trust it. And so it's just a puzzle piece in the validation of these drugs. And so specifically when you know what you're looking for or the pathway it acts on, you would use the model that has that defined functionality.
Got it. Yeah, that makes sense.
And so, getting the gene expression when protein expression closer is, definitely the goal, but even from different donors, you have different variations. There's always, and it does have we always want to mature these tissues even more.
Yeah, makes sense. And it looks like we have a question also from the chat. I'll try to just maybe read it. So it says, "Hi Kacey, great work. The microtissues you've matured are impressively large. Have you looked into necrotic core / cell death at the interior? And then also on this note, did you have a lot of optimization into getting the right density for each tissue niche to get them into biofunctional, physiological relevant levels?"
Yeah, we're pretty much at the maximum size where we don't get a necrotic core. But during the maturation phase, we culture these tissues in a lot of media. So the cardiac tissues are sitting in a huge dish of 50 mLs of media and beating. So you're getting nutrients in. But we, have very much optimized their size. And going larger, you would need to include vasculature. And going smaller, you tend to lose functionality or don't always form the same way. And so it was kind of that sweet balance of, what gives us the most functional tissue at the end of the day, robustly. So we looked at what was upregulated in the mix, and you'll see you're activating, more stem cell pathways, notch signaling, keratin. It's Wnt signaling, so they're becoming more stem-like and plastic, and they are stem cells, so that would be expected. And if we do gene ontology for the cellular components in the heart, we're really losing everything in the mixed condition that we worked so hard for. So everything related to contractility, T-tubules, AM band, sarcomere, etc. So now we wanted to look into tissue specificity. So we cryo-injured the heart, and we looked at a specific immune infiltration only into the heart. So in the interorgan group, you can see that the immune cells go directly to the heart and not really the other tissues, whereas in the mixed condition, they're going all over the place. And this is replicated with troponin secretion. So we're not getting that localized response in the mixed group, but when we have the interorgan vascularized, we are. This is what it looks like for a longer period of time. And then we also asked if we did a healthy heart next to an injured heart, just to make sure, immune cells didn't just love the heart and only go there. And it really just went to the site of injury, not necessarily a tissue. We looked into physiological PK/PD modeling. So with 4 tissues in the reactor or 1 tissue. So this was another thing from the reviewers, using the right controls, which was a hard review to get, like, oh, you didn't think I used the right controls. So their point was that we were comparing to tissues in isolation, whereas we needed to decouple the 4 tissues from each individual tissue with and without the vasculature. And so we went back and we did that, and we made a PK/PD model for that as well, but allowing you to parse out the effects of the tissues versus how we're connecting them versus the vasculature component. We see that the interorgan group kind of lowers that peak doxorubicin bolus. So we're now delivering doxorubicin into the system, and we're picking this the maximum amount or a defined dose that induces heart failure in most patients once they hit this dose. So kind of really stressing the system to see that response. In the mixed group, because you have no vasculature, you have a higher peak value, which is not physiological, but in the interorgan group, we see that it is, and you get the tissue-specific responses, so decreased albumin in the liver, injury in the heart, etc. When we look into the clinical relevance of this, we looked into microRNA bioreactors because we can compare to these clinically. And first looking at cardiotoxicity. So that's that initial injury that's showing initial damage to the heart. And MIR1 is kind of the only agreed upon biomarker at the moment that's predictive. And we see that only in the interorgan group, whether we have 4 tissues, just the heart in the 1 tissue configuration, or we're pooling from the perfusate, so mimicking like a blood sample, only the interorgan group are you seeing that MIR1. And then when we look into cardiomyopathy, so kind of a later stage onset of heart failure, the interorgan groups really cluster really nicely with the clinical data. And you can see in the bar graph at the right, looking at the top microRNAs that are predictive to future development of heart failure, cardiomyopathy. The interorgan 4 tissue group kind of scores the best at being both sensitive and specific for predicting this response. So it does have some clinical relevance when benchmarking. And so what method was critical to complete this paper and the technique here that's novel? Mostly it was how we...
Kacey, before you get
Yep.
too much into this, we have another question from the chat, actually kind of related to the question I had about maturation.
Yeah.
So I guess the question is basically getting at so there are differences between the iPSC-derived tissues and adult, human tissues. Do you have an idea of what specific genes are different and how you could kind of induce additional maturation in those tissues?
Yeah, there are a good amount of genes, so mostly related to their metabolism, their contractility, calcium handling, and then the ion channels. So one of the main things you're trying to drive out in IPS cardiomyocytes is this funny current. So IPS cardiomyocytes spontaneously beat, and if you take an adult cardiomyocyte, you wouldn't see that. And so they have this funny current. I didn't name it. That's a bit overactive, so trying to shut that down, and then just a switch in metabolism to more oxidative versus glycolytic. So being able to use fatty acids is important. And there are a handful, and it'll definitely depend on, if your drug's affecting ion channels versus contractive force. The main ones are, force has not been there, so things related to myosin binding proteins, your sarcomeres, your myosin light chain versus heavy chains. And so we don't really know how to get them there. We're getting them closer for sure. And you have different ion channels in animals, so it's just nice to have a human version. And then you don't have human cardiomyocytes that you can ever get. So this is kind of where we are for now. And we just keep trying.
I suppose knowing how to get there is the next Nature paper.
Yeah, I mean, there are a lot of efforts around what's in the media to mature them, and most of that's making metabolic switches happen to kind of force them along the way. But having the right stromal cells and making them biomimetically do whatever their function is kind of coaxes them along. So we could leave them in culture for longer and they get better, but hopefully there'll be more pathways we can activate to kind of coax them along the way. But there's definitely a lot of people working on it in the heart and the liver the most. So our novel method, which at the end of the day, if you think about it, you just use the trans. Well, is it that novel? But it really helped our system work. And so that was just the novel thing, is how we connected organs. And so there's been this push to have a whole body on a chip, all the organs connected, and you would have media routing different ways depending on what that organ saw in vivo. And our approach, so how it's done in the field commonly is just putting them in the same petri dish. So static culture, they're sharing a common media, or you're recirculating this common media. And during the tissue chip 1.0 phase, there were supplement funding to try to find a common media, but there wasn't really a common media that satisfied everybody's needs and tissues. And so we stopped trying to find a common media and instead looked at having our own optimized media, especially because a lot of people are optimizing the media and chemical components, small molecules to drive maturation. We want to keep that in the media and not have this common media where we're diluting and getting rid of all our maturation. So having the vasculature barrier with this tissue specific separation. So just having the vasculature underneath. And so why did we get to such a simple approach? Well, we tried alternative approaches, and I'll kind of walk you through them. So our original goal was, of course, we're going to vascularize the tissues and flow through them. And then we'll have media exchange in each well. So still have these niches because we want the tissues in their ideal environment, but we're also going to flow through them because this just really mimics in vivo physiology. So this was our approach. And so on the left, this was our reactor. So we're flowing through the tissue and around the tissue. And then for scaling and organ order, you can put a couple skin with a much more liver, or skin's your biggest organ. So 4 livers with 6 skins with 2 hearts, and just change the order and the scaling to kind of address those problems. We weren't working on that because there are big problems to address. What are the ratios for different organs, but staying open. So this is our video, what our platform looked like, and then we got to work on that. So we, had to incorporate perfusible vasculature into our tissues. So this is our cardiac pillar system. We made them hollow. You can see, we can perfuse it. We're basically using sutures to form these cardiac pillars, and we had L-shaped sutures because as you would pull the suture out, you would get like a flick at the end that would break your tissue. So a lot of optimization went into this hollow tissue. The skin already had methodology using a sacrificial alginate network. That wasn't great for the cardiac tissues because dissolving alginate can be tough, and the heart is really sensitive. In the bone, that's a hard, porous scaffold, so vascularizing that was difficult. And so we looked at dialysis tubing, but everything was too big and not small enough. So we used PTFE, which is basically a transwell, and we micromolded it to fit into this, Pac-Man, that's what we used to call it, C-shaped bone, and have an inlet and an outlet. And then it's like great in practice. We could perfuse through the tissues. This one went really fast. This one went slower because you have pressure buildups. And so this is like an actual tissue, and those pressure buildups, you'll just see everything starts to leak out. So sometimes it worked great, and other times we just get these right angles were not ideal. And so then we kind of took a step back and thought about what do we really want from integration of tissues? And we weren't looking at problems related to the microvasculature. We just wanted them to systemically talk, because in things like systemic disease, mostly fibrosis or inflammation or other systemic diseases, you have a crosstalk of secreted factors per se. And so we weren't focused on microvasculature diseases at that tissue vasculature interface. And so we could just unroll the vasculature and put it at the bottom and have this planar configuration. So you'll see our videos here. And it's much easier to make, and it lets the tissue still crosstalk. So it's our first planar flow bioreactor. We've got all this tubing. We have like our own perfusion pump system, and lots of tissues and reservoirs. Our second one, we
got a quick question for you, if you don't mind. Sorry to interrupt. So you have all these kind of like perfused tissue models. It looks like that was probably a lot of fun to kind of like go through all those, but it's also a lot of work that didn't end up in the paper. Are those still like being used at all in the lab for other, I mean, they could have potentially other applications, of course, or are those things that are kind of just like, well, we tried it, it didn't work, on to the next thing?
Yeah, I mean, more the latter. So the vascularized skin tissues Angela Cristiano's lab has really used. We keep wanting to use the hollow cardiac pillars for different things, but we just haven't at the moment. It was a lot of work to make these beautiful hollow pillars, and so if anybody wants to use them, they're there. And then putting a vasculature in the bone the way we were doing with the membrane, that kind of actually led to our flattening it out. So we didn't really revisit that one, but we had a lot of milestones for this grant. And so we had to just try and, throw darts at the wall, see what stuck, and not get too attached to anything, because at the end of the day, we just needed things to work, and you keep moving, or so some of these were just done in parallel. And the simplifications mostly came from, you want it to answer the question and have complexity in that end, but not be so complex that you're getting all this variability every time and you have a lot of failures. And so kind of balancing those. And we like to just try new things and just look for whatever will work without getting attached. So we have like a graveyard of bioreactors. We always joke we should just put it on display, because I'm showing you some of them, but there are a lot. This is our second one, where we're flowing through this membrane. We have tissue specific media on the top, and we have this one big membrane on the bottom. And it worked well, but it wasn't robust, particularly because we were coupling everything to each other. So if we had one failure, the whole chip was done. And so we adapted to this modular workflow. So you've got these modular transwells, all our tissues, instead of having like the cardiac tissues in sets of 3, because we're always like, an N of 3, that we'll just do everything in 3. Breaking them up into modular tissues let us have tighter quality control screening. So we could really make sure this tissue had all the functional metrics that we would want. And if it didn't, we didn't have to sacrifice everything else in that strip. We just got rid of that one tissue. And as we got better at making tissues, we didn't really have failures, but in the beginning, maybe half our tissues formed and matured properly. And so it was nice to have that modular ability. And then we also had made our reservoirs open because we were getting bubbles, and bubbles in a microfluidic system will just, shear everything off and ruin everything. You can't image. It's a nightmare. And so you'll usually do like coatings to get rid of bubbles, but we are open on the top anyway. So we just made a reservoir to serve as a bubble trap, and easy peasy, got rid of that. So this is what it looks like. And we were using this one for a really long time. And then I was presenting it at a poster conference at a Keystone. Somebody, brought up the point that, it's not a true N of 3 in this system. They're, just technical replicates. So good point. So we moved to a true N of 1, and that's how you got the strip. And we also tried to make it imaging compatible. So 4 of our glass slide strips will have the footprint of your typical micro well plate. We're good? So how has this paper changed since the original submission? So we didn't submit until we had that final design, but most of the work was, getting the bioreactor right. But the things that the reviewers really wanted was to prove inter-organ communication, so that ubiquitous tissue marker. They didn't like our supernatant base, like we were looking at different cytokines and things secreted by cells, and so we did the exosome label. They hated the term inter-organ. We loved it. We thought, like, we worked a lot on our naming. So also these tissue chip groups, they all had great names for their bioreactors, and we really didn't. So our tissue chip 1.0 name was Helva for heart, liver, vasculature, and we used to joke it's one hell of a platform. But everybody else had really unique, great names. That's all we could come up with. We mostly call our platform UH3, which is a grant name. It's like calling your project R01. Like but when we finally, Naveed on the team came up with inter-organ, and we loved it. Reviewers hated it though, so we had to get rid of that, but I still used it in this talk because I like it. They wanted more vasculature characterization and baseline tissue data. They asked for the PK/PD model, which, makes sense. And then using the right controls, which was hard, to read. The reviewers didn't think I did the right controls, but they were right. So they wanted us to parse out and do everything, each tissue in isolation with and without the vasculature. And, the real reason we hadn't done this before is that's a lot of pumps, and so we had to buy more pumps, and this was what it looks like just for the cardiac version. And so we also had reactors for the liver and skin and bone, and it just got to a lot. And so this is what one experiment looks like. You have to have all these different reactors because you have to have that isolated condition for each tissue. And so doing the data points, we go from the day all the way to the night. So in the bottom right, that's not the sun going down, that's the sun going up, because you have to do all the reactors at once. And so having all those right controls, was great, and the reviewer was absolutely right. But and we had to make all these platforms. So it was a lot of effort during COVID, and this is during COVID, the revision. So you can see we have our masks on, but just wanted to show what it looked like in the lab. So the next experiment, good.
That's pretty amazing to see. I always love seeing pictures like this. It seems like a lot of people have these, and you don't really get to see them very often. But so how many people are like working? I mean, this is like what, 100 plates or something? How many people are working on this at one time?
So it's me in the hood, Yoga right there, and then in the other room were Naveed and Emu, and we had to basically beg to have that many people in lab at that time. Like, we'll be separate, and we'll never be near each other for that long. Only 2 of us would, but because it was COVID, and so somebody took the picture, so we are more than 2 people in the room. But
yeah, no, that's
they're far away.
To see all of that together.
Yeah, it was an effort, especially during COVID, but it is nice to see. And so now you see that there's all this tubing, and that's kind of a pain. So our next experiment is getting rid of the tubing and putting the pumps on board. And so trying, this platform works really well in our hand, but we want to make it more user-friendly. And so having it in a well plate format with more universal compatibility, and then automated handling and sampling. So somebody at pharma could do their assay with these automated systems, but also us in the lab with our micropipetters. So we want to make it, usable for high-end users that have fancy equipment, but not forget, our roots and us in the lab where we never have robots, we just have us. So one of the tissue chip goals was to automate everything. And so people would have robots and we would say like, we're robot compatible, but in real life, robots are us, and like we are the robots. And also maintaining that open modular style. So you can just plug and play what you want, your best model, and not really forcing someone to change their design constraints. And then the final experiment or the future, we're constantly evolving. Hopefully you'll see new work from our lab where we have different tissue models going in and out. But basically, if you can mature iPS tissues, always keep working on the maturity. You need to connect them in a way that maintains that biological fidelity. So hopefully we'll have, every patient on a dish to either just get drugs to market faster, especially newer drugs coming to market. They're biologics, they're cell therapy, gene therapy, things, or antibodies, things like that, things that need human systems for testing, or large primates. And you can't always do that, especially not quickly. And so having this human tool where you can really get drugs to market faster. And then also taking in patient diversity with clinical trial on a chip. And so actually having personalized organ on a chips, in the lab, bringing in different ethnicities, different sex differences, different disease genetic mutations, and being able to parse these out and do these rapid clinical trials or see what patient should get which drug. So the right patient, the right dose, the right time, and really optimizing all of that. So patients are benefiting versus, having this drug development process that's just so costly and time consuming. And that's it. So.
Yeah, that's great. I think maybe we have time for a couple more questions, and maybe we can kind of cover some of this big picture stuff on this slide. So are there any specific things to kind of further bridge that gap between, animal models are still being used. So what needs to be done for like these types of human-based, human cell-based technologies to kind of eliminate some of the animal testing? Are those like kind of ongoing conversations with these funders, or do you guys have like outside funding too that's interested in this?
It's ongoing. So the stakeholders, being either the FDA or different pharma companies, are very active in saying what they would want to see. But it's not, so the shift in these tissue chips, at first it was, replacing animal models, refine, replace, and then the next word is reduce. And so organs on a chip or these human situations let you reduce the amount of animal studies you do, but there's not, you're not going to get all the complexity of an animal model. And so it's a nice tool to bridge the gap and not necessarily always replace them. Or you might want to screen, instead of screening first in animals all the time, you can screen first in this human setting, especially if you really know your disease mechanism. You can kind of parse it out and do mechanistic studies easier in an organ on a chip, because I can knock out most things in my chip versus in an animal. I can't just go around, doing any genetic modification I want and expect that mouse to survive. So I have a bit more control to parse things out. And then newer drugs, the animals won't even express the right receptor. So you have to have a humanized version and then make the animal version, and it's just back and forth. So it's just a nice addition to the toolbox. And they've been used. The FDA, or the government, actually signed passed a law recently that you don't need animal data to get a drug to file an IND. And so organ on a chip data can, so it just, not saying they don't want animal data. They want all the right data to prove that the drug is safe, whatever context that is. But it's not always going to be animal data. And so using other data is just fine and acceptable when the supporting data is there. And so Hesperos, an organ on a chip company, they actually got a drug straight to IND to start going into trials using their organ on a chip. And so there are certain use cases where there's just not a good model at all, and this is the best situation, or somewhere it's telling you something that animal models aren't, or in the later stage, once it gets to clinical trials, a lot of times they'll go back and discover, oh, this drug shouldn't have failed overall, it just was given to, the wrong patients. And so if we narrow down our clinical trial and only give it to these patients, it would benefit much more. And it seems, intuitive to only give the drug to patients that it would work the best for, but that hasn't been done in drug development's been very much like dig through the dirt, like find molecules, run them in a screen, find a hit, and then give them to everyone and see how everyone does. It wasn't until Keytruda that they actually said, okay, only patients with this biomarker mutation should get this drug. And people thought they were crazy. Like, why would you limit who you can give your drug to? But in doing that, they got their drug to market and captured the whole market by being specific, because it worked, because they only gave it to the people who would respond. And so there's been a big shift in designing drugs for specific people versus for the masses. And you saw in like the beginning where most of these top 10 drugs, they barely work for anyone, like max 25%. So that's wild. So we need to personalize medicine. Like I can get clothes to fit in any which way, any style, anything I want, but my medicine options are, very global for the masses. There's no personalization.
Right. Have you guys started comparing like iPSCs from different patients and identifying some differences there? I mean, that's obviously like each one of these is a ton of work, especially when you can only have, 3 people or whatever working on it. But have you started doing those comparisons?
Yeah, we've been doing so male and female, just cell lines, plus and minus the corresponding hormones, and then different genetic mutations that are known to cause a predisposition. So not just doing these global, because we don't have the manpower to just have like huge IPS banks that are representative of the population, but we do know that certain, especially in the heart, we do have a family history of heart disease as a question, because your genetics do play a role. And so we can pick mutations and use iPS cells with those various mutations that are known to have a risk. And so kind of using population-based statistics to choose which patients we should model, since we can't model everyone or even a majority at the moment.
Yeah. And I guess that kind of leads into another question about just a very practical question. So how long would it take to take a patient's cells, reprogram them, make all these tissues, and that basically make one of these chips?
So the differentiations are, like 2 weeks, some more for the other tissues, but let's say a month for differentiation, a month for maturation, and then you'll connect the tissues for a month. So maybe 3 months to do your whole tissue chip study, but your iPS derivation takes a while. So generating the iPS cells, picking the best clone, and then passaging them up to a passage number where they differentiate well. So like maybe 3 months at best, but sometimes it can take longer. And each iPS line, you kind of have to change. So with cardiac differentiations, you're using small molecules. You'll change the concentrations based on, that cell line, and also sometimes that passage number, your matrigel lot. Like there are a lot of factors that go in, and then especially during the pandemic, tissue culture plates were changing all the time. Like everything's changing, and so the system can be a bit more chaotic than any of us would ever want. And so having quality control measures that actually say, what is a good cell that's suitable, and then putting that into a tissue, and what's a good tissue that's suitable, et cetera. We keep trying to refine. But the big problem, like making iPS cells from each patient, takes a lot of time to have a precision medicine workflow at the moment. And so we've been, having just iPS lines that we normally use that are kind of representative in a small context. And at least we know we can rely on their results, and then plugging in patient either immune cells or for cancer studies, their cancer cells or their mutation. Like we can put their mutation in to the cell lines that we have versus having a different donor all the time. So, but it all would depend on the question you're trying to ask. But yeah, having big IPS banks is not something we're capable of. And we have a stem cell core that we built out with this grant to kind of do that. And they have a good amount of capability, and so they generate the iPS cells for us, but it's a lot, and it's a lot of money.
Yeah, for sure. This is, I mean, this is really, really amazing work. Thanks a lot for joining us today. I think Bria is going to maybe say a couple words to close us out, but this has been really awesome. Thanks.
Yeah, this was amazing. I mean, I think Michael said it too. That picture of all the flasks and the plates like that, that's the stuff that we like to see. So yeah, just kind of knowing what went into the work. So yeah, thank you so much for sharing your data, and as well as the story behind everything. Thank you to everyone who is on the Twitch live stream right now. This video should be posted on our YouTube in a couple of days. So if you missed it, or you missed a part, or you want to send it out, please check out our YouTube. And so with that, we'll stop the stream.